Hey everyone, this is Jen, and this is the re-recording of the flavor discussion from our Aroma Explore the Wheel Pome and Melon webinar. As you may have recalled, I have had some audio issues the last time I tried to present this, and I'm happy to say that the issue turned out to be me the whole time, and I think I know now what I'm doing, and at least I know not to do what I did again. So I didn't re-record the kind of the front matter of the webinar or towards the end because I know that's when you all were able to hear me. So we're going to pretend doodly 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 doo. We've done our warm-up exercise. We have done our first 5-minute blind smelling panel. And now we're going to move on to our flavor discussion. So far, in our webinars, we've talked about malt and hops, and today we're focusing on yeast and specifically on esters. When we're talking about yeast and fermentation, it's important to remember that yeast is simply trying to survive in a stressful environment. It does not give AF about your desired flavor profile or the finished beer. And you'll hear me mention that a few more times because I know, particularly for brewers, we tend to almost like personify our yeast, kind of give them a personality. They definitely do have personalities, but they're not going into the wort thinking, how can I make this beer the very best beer that Jen has ever brewed, right? The yeast is like, what is happening? I am so stressed out and I'm just trying to survive. So when we're talking about yeast and fermentation, there's going to be 3 main stages of fermentation: the lag phase, the active phase, which you may also see called the log phase, and the stationary phase. Now, when yeast will create flavor compounds, there's 4 main groups that those fall into: esters, fusel alcohols, off flavors, and phenols. And yeast creates these flavor profiles for one of 2 reasons. Either it's trying to remove something toxic, or it is a byproduct of an essential process. And esters are made by yeast proliferation and detoxification. So esters are going to be made when yeast is trying to grow, trying to proliferate, right? Trying to clone itself. And also when yeast is trying to detoxify what's going on around it so it will be able to survive. So when we're talking about esters, these are going to be one of those groups, one of the 4 main groups of flavor compounds that yeast will form during fermentation. And an ester is a volatile compound formed by an organic acid and an alcohol. It's the largest group of flavor compounds in beers, and a lot of times it's going to be responsible for those fruity and floral flavors. That is a mouthful to say all at once. The way to remember your esters, where they come from, is the alcohol is going to be the first name of your ester. The acid is going to be the last name of your ester. So let's take ethyl butyrate, for example. Ethanol is going to be the alcohol, and butyric acid is the acid. So when those 2 are esterified, they become ethyl butyrate. Another way you might remember this, I learned this from Richard Price at Escarpment Labs, is Esther's a really fun girl. She goes to parties and drinks alcohol and does acid. Whatever way works for you, that's the naming convention. And for me, I find it very helpful to remember what this naming convention is, particularly as we're moving through this discussion of esterification, which I'm never quite sure if I'm finished saying that word or not. But just keep that in mind, because that's going to help you kind of connect the dots between what's happening during these processes. So esters are commonly found in nature, in flowers and fruit. And a lot of times we might say, let's say we'll use ethyl butyrate again as an example. Ethyl butyrate smells like overripe pineapple, overripe mango, tutti fruity, kind of, gum, like a candy sort of flavor. And not that tutti frutti is a product of nature, but with things like pineapple and mango, where actually it's more correct to say that esters those are the exact same esters, right? We're smelling the exact same compound, whether it's in a pineapple or whether it's in a beer. That aroma active compound is going to be the ester. And so yeast learn to mimic these flavors from nature so that other organisms will move them around, which I think is fascinating. And one of the jokes is that yeast has tricked humankind into keeping them alive in various ways. So, people are out here like worry about the Illuminati or a woke mob or something. And like, really, yeast has kind of been our puppet master for a very long time. And this last bullet point I put in here just because I wanted to make sure to include it somewhere. Transesterification occurs when an acid reacts with an ester. So remembering our ester is made up of an alcohol and an acid. This new acid comes in. It's going to react with the ester and it's going to replace that original acid within the ester. So transesterification is, kind of like a step parent. Sort of in that way. You find transesterification more in mixed fermentation beers, but I did want to include it just to make sure you were aware that that is a process that can occur. So let's talk about proliferation. Remember that yeast will make esters to proliferate or to detoxify. So yeast has a full range of metabolic pathways that include glycolysis, the Krebs cycle, aerobic respiration. Glycolysis is the first step in yeast metabolism. Glucose is one of the best sources of energy for yeast. Other sugars, such as maltose, which is the main sugar found in wort, will be converted into glucose before entering the glycolysis step. So during glycolysis, this is going to be our first step in yeast metabolism. This is when we're in the lag phase because yeast isn't actually actively fermenting right now. It's gearing up to start converting those all of those wort sugars into ethanol and carbon dioxide. So during glycolysis, this glucose molecule we have is split in half. And when it's split in half, it creates two molecules of pyruvate and it releases energy from that broken atomic bond between the glucose molecules. So now we have two pyruvate molecules that can take two potential pathways. The first pathway is the fermentation pathway. Remember that glycolysis is when the yeast breaks glucose down into two molecules of pyruvate and releases energy. So we've got these two molecules of pyruvate. So during the fermentation pathway, those two pyruvate molecules can be converted into carbon dioxide and acetaldehyde. And if you're familiar with acetaldehyde, you know that that is the penultimate step before yeast creates ethanol. So if you have a healthy yeast strain, if you have enough viable yeast, that acetaldehyde will be converted into ethanol. During that conversion, that's going to lead to the regeneration of electron accepting molecules, which is going to then allow further rounds of glycolysis to continue. So this pathway is always active if enough glucose is present, even if oxygen is present. So, we'll talk about the role oxygen plays at the beginning, the aeration of the wort. And then, kind of after that step, we're really trying to limit the oxygen ingress into the beer because that's going to, it can age the beer. Oxygen after a certain point is not going to be good for your beer stability or your beer flavor. But at the very beginning of your fermentation, oxygen is very important. And I say that because this fermentation pathway, if oxygen is present, it will still ferment. You're just going to get a lot of undesirable off flavors in your wort because there's too much oxygen. Whereas what we're wanting yeast to do is we're giving it just enough oxygen to be able to proliferate, to be able to grow. And that way we can control those off flavors. Some pyruvate, instead of going through this fermentation pathway, will instead undergo a series of reactions that are going to convert it into carbon dioxide and acetyl-CoA. And acetyl-CoA is an acid. The CoA is coenzyme A. For our purposes, I'm just going to keep saying acetyl-CoA. Just remember that that is an acid. So pyruvate, like I mentioned, that isn't used for glycolysis will be converted into that acetyl-CoA and carbon dioxide. Once acetyl-CoA is formed, it can take one of three pathways. So we have a lot of pathways happening in each one of these steps. The three pathways that this acetyl-CoA can take is the Krebs cycle, fatty acid synthesis, and esterification. So the first pathway it can take is the Krebs cycle. And the Krebs cycle requires oxygen. So this is one of the steps where oxygen is very important to have. During the Krebs cycle, we're producing intermediate compounds that are important to amino acid and fatty acid synthesis. And in the Krebs cycle, that acetyl-CoA is broken down to provide energy. Once it's broken down, this forms large quantities of ATP, which provides energy for various processes. So the Krebs cycle is, it's an intermediate step in fermentation. And actually humans also, this is how we create energy, is through the Krebs cycle, which is why as humans, we need a steady supply of oxygen. So that way we can continue this Krebs cycle so we don't die. So then the next pathway that acetyl-CoA can take is fatty acid synthesis. This also requires oxygen. So this acetyl-CoA that was not used for glycolysis, it wasn't used in the Krebs cycle, is going to be used here to build long-chain fatty acids. Those fatty acids combine to form lipids, and then those lipids are going to help form our cell membranes. Lipids are a required building block for cell membranes. Without oxygen, yeast will attempt to proliferate this way, but it will have fewer building blocks. Because it will have fewer building blocks, of course, those building blocks being our lipids that were formed by our fatty acids, that were formed by our acetyl-CoA, our cells are going to be smaller and our cell membranes are going to be weaker. They're weaker in the way of being more porous. Being more porous means they can leak a variety of compounds into beer, creating off flavors. And this, one way I kind of think about this with this fatty acid synthesis, if you don't have strong enough cell walls, is that it's similar to what happens when yeast dies, autolysis, right? Autolysis is literally self-death. And when yeast dies, we say it dies because those cell walls break down and weaken, and the cells kind of burst open and just kind of spill their guts into the finished beer. So if you don't have enough oxygen to create these healthy cell membranes, then you're going to have, that's going to happen way before autolysis will happen. So then the last pathway that acetyl-CoA can take is esterification. Esterification does not require oxygen. And this happens because it is possible for an overabundance of acetyl-CoA to leak out of the cell during fermentation. So let's say that maybe we're doing the Krebs cycle, maybe as fatty acid synthesis, and it's similar to when you have alpha-acetolactate that leaks out of your yeast cell and then oxidizes into diacetyl outside of the cell. This is kind of a similar thing. So you can have an overabundance of acetyl-CoA that's going to leak out of the cell, and then it's just in the wort. And the issue with this is that our coenzyme A, the CoA in acetyl-CoA, is vital for transportation of acetyl groups and fatty acids within the cell. It's a limited resource. And because it's a limited resource and yeast has to have it, it cannot afford to have that CoA, that coenzyme A, floating around outside the cell. So that coenzyme A is going to be scavenged back into the cell. And then that's going to happen through this process of esterification, which is where our acetyl-CoA is going to be catalyzed by enzymes known as esterases. I will not say it the same way twice in this entire presentation. So remember, when you see the suffix ASE, you're dealing with an enzyme. If you see ATE, like ethyl acetate, that indicates a polyatomic anion, which is a negatively charged anion. So we see esterases, that tells us we're talking about enzymes. So alcohol acetyltransferase, or AAT, is a special membrane-bound enzyme. When I say membrane-bound enzyme, that means, really just kind of exactly what it sounds like, right? So you've got this enzyme. I kind of think of it as like little fingers sticking out from your yeast cell, and those are going to reclaim that coenzyme A by breaking that acetyl-CoA to ethanol. So it's going to, again, there are AAT is an enzyme that's going to kind of sticky fingers grab this acetyl-CoA that might be floating around outside the cell, because it's going to break it down to get that coenzyme A back into the yeast cell where it is much needed. It's vital. And then we're left with this ethyl acetate, we're left with ethanol. So this breakdown leads to the most common ester found in beer, which is ethyl acetate. So again, ethyl coming from ethanol, that's our acid. Acetic acid is what's going to form that acetate. So they're going to esterify together into the ester known as ethyl acetate. So AAT and other esterases scavenge that coenzyme A and break it down into esters. Esterase breakdown depends on yeast strain and species. Different strains will have slightly different esterases, which means that, you might have one yeast strain that produces a lot of ethyl hexanoate, a lot of that candied apple kind of anise sort of flavored ester. You might have another yeast strain that's going to create a lot of isoamyl acetate, right? That artificial banana flavor. So different species, different strains of yeast will have different esterases, and within those strains, they will have different levels of preference for producing esters. So I like to think about that as, maybe your parents say you have to eat your vegetables before you have dessert. Maybe somebody else's parents said eat dessert whenever you want. You can have vegetables later or not at all, right? So there's different levels of preference. And this is one of the reasons why your yeast strain, when you're brewing to style, or if you're just looking for a particular flavor profile, you're going to want to see what kind of esters your yeast strain can create. So that's esterification. Now let's talk about the other side, remembering that yeast will make flavor compounds to either proliferate or to detoxify. And that brings us to fusel alcohols. You may find yourself wondering, like I did, where other alcohols besides ethanol come from? Because if alcohol is the first name, then why don't all esters start with ethyl? And that's because esters don't only come from metabolic processes. They can also form when a fusel alcohol reacts with acetyl-CoA. So fusel alcohols are an important precursor for many esters. They're very commonly found in beer with stressed fermentation, particularly ones with inadequate free amino nitrogen or FAN. Fusel alcohols are byproducts of the degradation of amino acids needed for cell growth and repair. So again, this is a byproduct of yeast trying to survive. So our fusel alcohols are formed through the Ehrlich pathway. What happens in this pathway is that amino acids are stripped of their nitrogen. They're stripped of their nitrogen so they can produce other amino acids, so they can produce DNA and other essential compounds. Stripping results in alpha keto acids. Now, these are toxic to the yeast cell. The yeast needs to detoxify. It needs to get this fusel aldehyde, this alpha keto acid. It needs to get it out of the yeast cell. So it has to break it down. Alpha keto acids are broken down through two steps. CO2 removal will produce a fusel aldehyde, which is also toxic to yeast cells. So we remove the CO2 and it's like, well, okay, now we have a fusel aldehyde. That's not an alpha keto acid, but it's still toxic to us. So that needs to be broken down further. The fusel aldehyde is reduced via NADH, which produces a fusel alcohol. NADH is abundant in properly oxygenated yeast cells. So again, here's another area where oxygen pops up and the importance of aeration. I think particularly this discussion of esters really demonstrates why we aerate our wort. If cells are not properly aerated, there's limited NADH, and that results in reduction not happening and fusel aldehydes remaining in the beer. So this Ehrlich pathway is also driven by amino acid synthesis. And this is one of those things, and I know I say all the time, like, one thing I find interesting about beer, but one thing I find interesting about beer is that there are still processes happening, in this year, 2025, that we still don't quite understand what's happening or why it's happening. And to me, I find that fascinating because it seems like, we can control so many parameters of creating a beer, but there are still just some things that we can say are magic. We just don't quite understand it yet. This is one of those things. It's unclear why yeast does this, but the general opinion is that it's possible that yeast does this to increase ester production. Again, remembering why yeast is creating esters, it's to mimic smells and flavors found in nature to be able to trick, fruit flies, bees, birds, whatever, into transporting yeast and helping it stay alive. So on this slide, this is a table of amino acids that are transformed into their respective fusel alcohols via that Ehrlich pathway we just talked about. Like I mentioned earlier, these fusel alcohols are where we get esters like isoamyl acetate and ethyl phenyl acetate. So I find this, I really don't think that there's, unless you're getting like your PhD in beer chemistry or something, there's not a world where you're going to need to like memorize which amino acids create which fusel alcohols. I just put this in here because for me, it was really helpful, again, to say like, well, wait a minute, where do other alcohols come from in the beer if it's not ethanol? And then why aren't all esters named ethyl something, right? So we can see here, something like leucine is going to create isoamyl alcohol. That isoamyl alcohol is going to react with acetic acid, and that's going to give us isoamyl acetate. First name, alcohol, last name, acid. So as a brewer, there are a lot of levers you can pull to control the amount of esters your beer has. The first one, obviously the most important one, is going to be your yeast, right? The yeast strain you choose is the greatest factor in determining ester production. If you're looking for, like a beer with a really high isoamyl acetate, that artificial banana flavor, you're not going to use a lager yeast for that. You're going to select the yeast strain that's going to give you those esters that you want. And just as importantly, will not give you esters that you don't want. So if you're looking for more esters, 3 things you can do, well, really 2 things, is one, you can create a yeast starter. If you're not familiar with a yeast starter, this is something that I've done, I do every time I homebrew. It's also something that breweries will do, obviously on a much larger scale. But generally speaking, what we're talking about with a yeast starter is we're taking the yeast that we're using in our fermentation, we're adding it to a specific quantity of wort that is, I don't remember off the top of my head what the gravity is, but it's, it's a fairly like inoffensive gravity for the yeast to start its lag phase a little earlier and to start proliferating in this wort starter or in this yeast starter so that once it's added to your wort, it's ready to go. So think of a yeast starter as like, hey, yeast, we don't need to leave just now, but maybe start thinking about like getting your shoes on, finding your keys, all of that stuff. Fresh or freshly harvested yeast obviously is going to give you more esters because fresh yeast is going to have healthier cells. You can underpitch to create more esters. I don't know why you would do that. Typically, underpitching, if you have a lot of esters, underpitching can possibly be a culprit of your flavor profile not turning out the way you want it to. Underpitching your yeast is not the most efficient way to create esters, right? We have much better ways. But let's say if I'm judging a beer and I find it to be very estery for the style, one of the things I will suggest to somebody is, hey, check out your yeast, make sure you've got the viability and you've got the, the cell counts, that just check to make sure you're controlling as many of your parameters as possible around your fermentation. If you want less esters, you can use old yeast. And again, you can overpitch. Overpitching is really, really, really hard to do. For both of these, if you came to me as a brewer and you're like, hey, I want to brew beer, I don't want a lot of esters in it, using old yeast or overpitching are like the very last things I would tell you to do. Again, we just have more efficient methods, namely our yeast strain. So for temperature, this is another lever we can control. We can use to control our esters. If we want more esters, we might have a higher fermentation temperature. Higher temperature also encourages production of fusel alcohols. It can also affect the type of esters being produced. So again, this is something, let's say as me as a home brewer, if I'm making, let's say I'm going to make a juicy IPA, and so I'm selecting a yeast strain that's going to have a lot of those fruity flavors to it. I may also want to ferment it on the higher end of its recommended temperature range if I'm looking to maximize those esters. That's something that you might do. Again, a lot of times if I get a beer that's, if I'm judging or evaluating a beer and I get a beer that's seems very estery, seems like it's not supposed to have the esters in it that it does, particularly isoamyl acetate, that one's usually a pretty good giveaway that you've had a poor fermentation. Then one of the things I would suggest is make sure you're controlling your temperature as much as possible during fermentation. If you want less esters, lower temperature. And I think this also is a good connection to make between the differences of ale yeast and lager yeast. Ale yeast is fermented at higher temperatures. We're going to get more esters from ale yeast. If we're using, let's say, like an American strain, like the Chico strain, American ale strain, that is going to be very clean, very lager-like, then we're not going to expect that many esters. But we will still get more ester productions in an ale strain than we will in a lager strain. Lagers, of course, are fermented at a lower temperature, so they're not going to create as many esters. Now, lager yeast will create esters if it's stressed out enough, right? If the yeast isn't healthy, if your temperature is too high, basically all the same things. So you can have a very estery lager, but that's going to be due to issues during your fermentation. So oxygenation, this is another one that is very big. And I'll back up a bit because I know I keep mentioning aeration and I didn't specifically tell you where it goes in this process. So in our brewing process, once we've completed our boil and our whirlpool, we need to transfer that beer from the whirlpool to our fermenter. But that beer is very, very hot, right? So we have to cool it down. I'm sorry, at that point it's still wort. We're going to put it through a heat exchanger to cool it down to around the temperature where we want the yeast to ferment. If we put the yeast in when it was, still around like 185 degrees Fahrenheit, the yeast is going to die. And what yeast doesn't die is going to make some terrible flavors in your beer. So when we're knocking out from the whirlpool through our heat exchanger into our fermenter, this is where we're going to aerate. So again, for a home brewer, when I home brew, I have a little oxygen tank that I'll just put in, oxygenate my wort for a little bit. Sometimes you may see people just kind of shaking their carboys back and forth. That's to also get oxygen into the wort. A lot of times with commercial breweries, they will actually inject oxygen into the wort as it's knocking out through the heat exchanger. But either way, we're adding oxygen before or as the wort gets to the fermenter. And again, we're adding enough oxygen that we can have a healthy Krebs cycle, healthy fatty acid synthesis, and all of the other things we talked about that oxygen is important for during fermentation. So when enough oxygen is present, if you've properly oxygenated your wort, most acetyl-CoA production will be used for fatty acid metabolism. When oxygen is absent or there isn't enough oxygen, that excess acetyl-CoA will remain. That's going to increase the esters through the esterification process that we talked about. Wort composition also has a lot to do with your esters. Your all malt wort has higher free amino nitrogen and also has a higher nitrogen to carbon ratio, which is going to lead to more ester production. Remembering that that free amino nitrogen comes into play when we're talking about how fusel alcohols are formed. When we say all malt wort, that means that you're using, all malt. So you're using only malted grains. You're not using rice. You're not using corn. You're not using any kind of an adjunct. So adjunct wort is basically wort that has been kind of cut with something like corn, something like rice. And that adjunct wort, because we've taken a portion of that all malt out, that adjunct wort is going to have lower free amino nitrogen, which is going to lead to less ester production. Your fermentation vessel also matters. Open fermentation, that's a cool ship. There are places like Dovetail use open fermenters for all of their beers. I think this makes sense when you think about the construction of an open fermenter. It's going to be kind of a large square. It's going to be fairly shallow and definitely more shallow than your cylinder conical fermenters. So you've got more surface area that's exposed to the air, right? So you have more oxygen getting into that wort. So your open fermentation will produce more esters. Your cylindro-conical fermenters, CCVs, those are, the big stainless tanks you see with the conical bottoms in most breweries. Those also carboys are going to result in less esters. The reason for that partially being that hydrostatic pressure is going to suppress ester production. And then a couple of just miscellaneous things that you can use to control your esters. One, unsaturated fats inhibit esterases, which reduces ester production. Now, this is in here when we're talking about unsaturated fats. I'm talking about you are specifically adding something to your wort. I have not heard of a case where like your wort just unexpectedly has too much unsaturated fat in it. If you're adding things like peanuts, something like that into your beer, that can inhibit your ester production. Isovaleric acid, I don't love it, but I do feel a little bad for it because it's kind of in its own way. Isovaleric acid not only inhibits that alcohol transferase, that AAT esterase that we talked about, that's going to create ethyl acetate, but it also inhibits its own conversion into an ester. So, isovaleric acid, like obviously you don't really want it in your beer. It's not a particularly pleasant flavor. But yeah, I do feel a little bad for it, like you can't get out of your own way sort of thing. So that is going to wrap up our flavor discussion for our Pome and Melon Aroma Explore the Wheel webinar. I think I came back on with you all around this point. So you can, bloop, 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 pretend like you did your 3-minute panel and finish up with the rest of the webinar. I really appreciate your patience in me being able to get this re-recording to you. As always, any questions you have, feel free to reach out. Jen at undertheginfluence.beer. You probably already know all the ways you can get a hold of me for questions. So with that, I'm going to leave you and thank you again for participating in this webinar, and hopefully I will see you next time.